I'm very pleased and very honored to be here, although I'm a neighbor, but I don't have a chance to speak here before. So I decided to talk about the part of the work of my group, working in fact on work which is inspired by biology. We also work, as Kay just mentioned, about diffusion of proteins in membranes, so some aspects which will be maybe more physics, but I decided to show you how you can be inspired by biology and try to address some physics question and also help biology. So I will start with these movies. So first, I also mentioned that if you have a question, I prefer you to stop me. I will repeat the question, and I try to help you to understand my talk, right? So here we have different examples of cells. Like here, it's a cell of the immune system, which is moving. Here is a cell which is dividing, and here's the cell with many protrusions. And what I want to address as stress here is the fact that these cells really change shape. This one, when it's moving, you can see that the front of the cell is really ruffling, and you can see a lot of tubular structure behind it. You can see, obviously, that the cell is dividing, changing even topology and shape extensively. And you can see this is the edge of a cell, and the cell has many, like a snail, a lot of extension to fill its environment. So this typical scale will be like, let's say, 50 microns. And if we go to a smaller scale inside the cell, you can see here this is a compartment of the cell, and you can see that this compartment, which is this globular structure, is also able to create many small vesicular structure, like small sphere. And you can see that all this structure all together change shape constantly. And this shape change results from the interplay between the membrane, which is the structure which is surrounding the cell, and some polymer which sustain the membrane, which are called actin cytoskeleton. So in the cell have to exchange a lot of material constantly, either with the exterior or with the interior. And what this scheme is showing you is different compartments of the cell. Here would be the exterior. This would be the membrane which is surrounding the cell. You can see plenty of compartment, and I don't need you to know what they are, but all these compartments, as you can see, all these arrows correspond to possibility of exchange between the different compartments. And this is, again, a movie where you can see, again, this structure here, which is called the Golgi apparatus, and this is kind of a structure which helps for the traffic inside the cell. You can see that as a rail, if you like. And this structure, you can see, form many tiny vesicles, and you can see also tubular structure which are formed, and all these small vesicles or tubules are the material which is used to exchange between the compartments. So there is not much exchange done directly by diffusion, but a lot of the exchange is made by forming this tiny tubular or vesicular structure. And this requires to strongly deform membrane again. So this here what we have is a collection of structures that you can find in cell, which are these structures which help to transport molecules that could be protein, lipids, from one compartment to the other, or outside the cell. So you can see these different structures. This one, I will talk a little bit later, is called a clathrin vesicle, but basically you have different, let's say, spherical structure, and if you look at the scale, you can see that all the structures are of the order of, let's say, 100 nanometer diameter. You have also, as I mentioned, you can have some structure which look like very tiny tubes. You can see them here, for instance. And you can see this part of the cell, which is called the endoplasmic reticulum, is in fact a full tubular network, as you can see here. And the typical diameter of all this tubular structure is the order of 50 nanometer. And this is also as a structure where you can see also some tubules, or better here, which is also of the order of 50 nanometers. So it means that the cell, to induce or to be able to exchange protein from one part to the other, or with the exterior, or towards the exterior toward the interior, has to form structure which are highly curved because they have the order of, let's say, of the order of 50 or 100 nanometer. So you probably don't know what a membrane, or many of you don't know what a membrane is about. So the cell is surrounded, as I mentioned, by the plasma membrane. This plasma membrane is mainly made of lipids. So this is this structure, these molecules here. They contain many protein, which could be either inserted in the membrane, as you can see here, or bound to the membrane like here, or inserted like this one. And so they comport a lot of lipid, a lot of protein, and as you can imagine, you can see the membrane as a very heterogeneous material. And in addition, and below the membrane, you can see this kind of a structure, which is the polymer I mentioned before, this actin cytoskeleton. This actin cytoskeleton interact very strongly with the membrane, and in a sense is able to create some of confinement of, let's say, of the protein or the lipids between this structure, like a fence, right? So I would say this is a kind of this is actually the actual view of the plasma membrane that people have right now. So to be back to the basis, I mean, as mentioned, the main component of the plasma membrane is lipid. So a lipid is like you can imagine a surfactant. It's made of molecules which are amphiphilic, meaning they have hydrophilic head group and an hydrophobic chain, like here. And if they are in water, like your soap, basically, they are able to self-assemble into a bilayer. You can see here, this would be the lipid bilayer. And this lipid bilayer has a typical thickness of 5 nanometers. So you have to keep in mind that your cells, all your cells, are surrounded by a structure which is self-assembled. There is no polymerization, no entanglement. So it's a kind of a self-assembled structure, and with a thickness of only 5 nanometers. So also something which is also important, you could just wonder what is the state of this assembly. And what I'm going to show you here is an artificial system made of lipids where a quantum dot has been attached to one of the lipids. And you can see this is basically real time. You can see that this quantum dot attached to a lipid is able to move, meaning that the membrane is fluid, right? So this is a simple phase diagram made to represent the different states of the lipid organization. And this lipid, you don't have to know what they are, but basically they will represent the more representative lipid of the plasma membrane. And if you make mixture of these 3 types of lipids, you can find different type of organization. The most common one, well, in this part is called liquid disordered phase. So it's like a, as you mentioned, disordered phase. There is also some organization which could be a little bit more ordered, and something which is absolutely not physiological is a case where you have only this type of lipid, and then you can have something which is a liquid crystal, like a gel phase. But most of the time, let's say the plasma membrane will correspond to this kind of composition here. And what I wanted to stress that if you look at the number here, which are mobility numbers measured with a technique called FCS, there will be the mobility of the lipid in this kind of organization. You can find a mobility of the order of a few microns square per second in this part when you have a liquid disordered phase. And you see that as soon you go in this direction towards the halo phase, you get a slower diffusion, but still you get some diffusion. And the plasma membrane typically has a diffusion coefficient at very small scale, which is of this order. But due to the confinement, due to the cytoskeleton, you might, if you look at larger scale, larger than the network, the mesh size of the network, you can find diffusion at larger scale, diffusion coefficient which are smaller. But if you want to keep something to compare to something you know, something you could compare to is the water viscosity. And the viscosity, the equivalent of the 3D viscosity of the plasma membrane will be of the order of 100 times the viscosity of water. So, but as I mentioned, what you have to remember, basically, the biological membrane will have this type of viscosity, so meaning that the membrane is fluid. Okay, so we have a structure which is self-assembled, fluid. So what is its mechanical property? So there is a very important property that you can already think about. First, if you have a material, you start to stretch it, right? So it's possible to stretch lipid membrane. And in this case, it's possible to define what is called the membrane tension, which is the variation of the stretching area versus the area of the membrane. And practically, to measure that, it's very convenient to use, and I will discuss this a little bit afterward a little bit more. It's very convenient to use model membrane system. I'm sure you heard about liposome already, but liposome are used a lot in cosmetic, for instance. But so practically, to measure the membrane tension and a lot of mechanical property of membrane, what people have used extensively is what is called giant liposome. So it's this kind of spherical, more or less spherical object made of a single lipid bilayer, you can see here. The typical dimension will be of the order of 10/10 of microns. And I remember you that the thickness of the bilayer will be 5 nanometer. And this would be an image that you can have with a phase contrast microscope. And you can see immediately that if you prepare this liposome with lipid and you look under the microscope, you can see that they spontaneously move. So it means that the thermal motion of the water molecule is enough to deform the membrane. So it tells you already something about the mechanics of the membrane, right? And as you can see here, you can also use some lipids which are fluorescent, and you can use a confocal microscope to look at this kind of structure. So if you are able to prepare that, after that, you can try to stretch it. And there is a very nice technique which has been developed, as you can see, not yet, and Evan Evans started to develop that in the 70s, but it's very elegant way to stretch a membrane. So what you do, you take your giant liposome, you take a micropipette, so this will be typically of the order, let's say 10 microns. You have something of the order of 5 microns. You take your micropipette, you aspirate. You use simply hydrostatic pressure for that. And what you can do, now you aspirate, you can measure the deformation. So you can make a very simple tension strain plot. And you can measure the deformation or the strain simply by image analysis. You measure the tongue length here. And if you do that on a simple lipids, and here this is a lipid I was showing on the phase diagram, you can see that you have a very simple elastic behavior. You can see that the tension is varying linearly with the extension for different type of composition. But you can see here there is a star. And it means that at that point, the membrane burst. And this is called membrane lysis. So you can imagine that this organization is made simply by simple assembly. And there is a maximum tension that the membrane can sustain. And after that, there is a hole formed in the membrane, and the membrane explodes. So it can be extended, but not much. And this tells you that your lipid membranes are very fragile. They can sustain a strain which is of the maximum of 5%. So this is one type of deformation. The other one, which is also very interesting, so if you think about, let's say, a balloon can be stretched but cannot be so much deformed in the other direction. Interestingly, the lipid bilayer, because they are fluid, they can also be deformed in the perpendicular direction, and they can be bent. And this is, again, something which has been extensively described by physicists in the 70s, especially Wolfgang Elfrich and Canham, who was a mathematician. And they were the first to propose an energy, a bending for the membrane. And they proposed that the bending energy would be proportional to the square of the mean curvature of the membrane. So this is mean curvature, and with a parameter which is called kappa. And kappa, this is, as any material, this is a curvature rigidity modulus of the membrane. And this parameter is a direct characteristic of a material. So it will depend directly on the lipid composition. And as I showed you before, you remember that the vesicle was able to spontaneously fluctuate because of the thermal motion of the water molecule. So this, again, with this type of techniques, for instance, with a micropipette aspiration, people have measured bending rigidity of the order of few kT up to 100 kT. So it means it's very easy, just the thermal energy is sufficient to deform plasma membrane. And just to give you a simple example, if you imagine that you have a plastic bag made of lipids, same thickness of the lipid bilayer, so then it will be 1,000 times easier to bend a lipid bilayer than a plastic bag of the same thickness, just to tell you how flexible it is. Okay, so I go back a little bit to biology. So I mentioned that the cells are able or have to exchange material with the exterior constantly. And there is an important process which is called endocytosis, which consists of internalizing molecules from the outside toward inside. As I mentioned before, to do that, you need to form a vesicle, and then to detach the vesicle and to have the vesicle, moving inside the cell. So this is different. It's not a snapshot, but different snapshots. It's not the same vesicle, but different snapshot of electron microscopy, which have been taken at the plasma membrane. And you can see that to have one of these vesicle form at the plasma membrane, you have different steps. You can see the membrane is originally almost flat, and gradually you can see that you have all this structure, which is very electron dense here, which is formed. Would be corresponding to this structure here. And you can see that gradually you form a spherical object, but this spherical object is still attached to the plasma membrane. So there are also some special proteins which are involved, which are able to cut the membrane here and eventually detach it. And when it's detached, if there is a cargo, this is supposed to represent a cargo, the cargo will be transported from here, the plasma membrane, toward the inside, right? And in this case, this electron-dense structure is this very nice organization that you can see here. This is kind of a 2D polymer called clathrin, which is forming a coat and somehow is able to stabilize the vesicle here. So in addition of the clathrin coat, there are also a series of proteins which are involved in the different steps of deformation, as you can see here. And you can see but you don't have to know it and don't care about the name, but you can see there is a series of proteins which have this very characteristic banana shape, which are represented here. All these green proteins, and all these green proteins are involved at different steps of the deformation. So these proteins, they belong to a very big family called the bar domain proteins. So they have this kind of very characteristic structure, which is forming kind of a banana or crescent, if you like. And they can be more or less curved. They can sometimes are very curved, and you have the scale here. And they can be less curved, and some have an inverted curvature. So it's a very large family of proteins which are involved in the deformation of the membrane. And if you look more closely to the same sketch, you can see that these proteins, which are shallow curvature, they play a role at the beginning of the process when the membrane or the neck here is not much curved. And interestingly, those who are the more curved, they bind at the late stage of the process, just before fission when the neck is very tiny. And the neck at that stage is about 10 nanometer, right? So the question is how, and the question we have addressed about the past year is to understand how bar domain proteins, this crescent-shaped protein, can bend membrane, and how they can also play a role in this endocytosis. So the most common way, if you care about how protein deform membrane, is to take small liposomes. So in this case, you can see the scale bar. It's a liposome about 50 nanometer, same thing, a single bilayer. You need some charged lipid because this protein needs charged lipid to interact with the membrane. You use a high concentration of protein, so for the details, macromolar typically. You incubate them and you do electron microscopy, and you can see that this protein, when they bind, they transform this spherical vesicle into tubular structure, as you can see here. Right. And then you can see the different type of proteins. So this one, shallow curvature, large tube. This one, more curved, narrow tube. And you can see when you have the inverted curvature, you can see that this would be a 3D images. And you can see in this case that the protein is inducing a tube towards the inside of the vesicles because it has an inverted curvature. So if you do the same thing with giant liposome, you can see that with an optical microscope, you can also see that this membrane spontaneously tubulate. And in this case, we don't exert any force to create these tubules. So now I have to go back to some simple, let's say, physics of membrane. So what you can immediately see on this different scheme that if you have a membrane which is initially flat, but interacting with a protein which has this, this kind of typical curved shape, like a crescent shape, if the protein is adhering strongly or is interacting strongly with the membrane and asymmetrically, so only on one side of the leaflet, you can imagine that this will, because the membrane is fluid, this will induce a spontaneous bending of the membrane, right? And it's the same thing if you have some structure which can insert between the head group of the lipids. They will also create like an edge. They will create also a spontaneous bending of the membrane. And this is called, in this case, spontaneous curvature. You can have exactly the same effect. Now, if you have some protein which are crossing the bilayer, but which have a conical shape, this, because of the asymmetry of the shape, you can also create some bending. Okay, so how does it reflect about the bending energy I was describing before? So in this case, so the bending energy is reduced because the membrane is spontaneously bent with this spontaneous curvature called C0. So, and you can imagine that C0, the more protein you have, the more bent membrane, right? So you can imagine easily that C0 should be directly proportional to the density of protein you have on the membrane. And at low density, so typically less than 10% surface fraction, you can even, and at least has been shown, you can even consider a simple coupling between the curvature of the membrane and the density of protein. And the coupling coefficient here is called intrinsic spontaneous curvature of the protein. So it's a parameter which somehow described locally, so it's like a coarse grain parameter in a sense, which describes how much the protein is deforming the membrane locally. So if you have a simple relationship between the curvature and the density, you see immediately that if the more protein you have, for sure, more bending you have. But in addition, if you have a membrane which is already curved, you see immediately that you will reduce the bending energy of the curved structure simply by increasing the density of protein. This is simply what this linear coupling means. So it means that if you have a curved membrane, you would expect to see an enrichment of protein in curved membrane. Okay? So if you are able to create, let's say you take a liposome and you are creating a curved structure like a tube, you can imagine that you expect that the higher the curvature, the more protein you will have on a curved structure as compared to the, let's say, the surface of a vesicle. So in this case, what you do, you minimize the bending energy because the more protein you have, the lower the bending energy. But something you have to take into account is the mixing entropy, because this will be at least there will be a cost, right? So in my group over the year, we have developed a technique which is somehow it's been designed to probe the curvature sensing of protein in particular. So for that, we have used a technique which is based on the formation of membrane nanotubes from giant liposomes. So we start from, you can see here, you have two different movies. This one with fluorescent molecules, and this one on a normal microscope. And you can see here the pipette, you can see a bead here, and I will explain how it works. So what we have is originally we have a vesicle which is adhering against the bead here. The bead is strapped with an optical tweezers, and what we do, we move the vesicle away using the macropipette, and you can see here much even better that, in fact, originally the membrane is deformed when you are going to move away, but since the membrane is fluid, at some point the energy cost is less when you form a nanotube because the membrane can be bent, as I explained. So you decrease the mechanical energy of the system by forming a very tiny membrane structure, which is like let's say a nanotube. And why do we call this nanotube? Typically because the radius of the tube you can form from a giant vesicle is of the order of, let's say, the radius, the smaller we can do is 7 nanometers. So not that far from carbon nanotube at the end of the day. And we can go up to 100 nanometer. And the beauty of this is, in fact, if you want to measure how a protein is enriched as a function of curvature, you need to change curvature. And here we have a very nice technique to to tune the curvature. So you can see here that the tube of a bare membrane, the radius of a bare membrane, is depending on the two parameters I was describing at the beginning. It depends on the binding rigidity, but it also depends on sigma, which is the membrane tension. I told you we can change sigma by micropipette aspiration. So you can see here, the more I aspirate, the higher the tension and the smaller the tube. So we have a technique to tune directly the curvature. Now we can inject protein with another pipette. The protein can be fluorescent, and you can look at the recruitment of the protein on the tube, on the vesicle. You can measure, you can do image analysis, and you can measure enrichment. We can also, from the lipid fluorescence, we can also look at the fluorescence of the tube to measure constriction. And if you want also, what is also interesting is you can also directly make mechanical measurement, because with the optical tweezer, you can measure the force in the presence of protein. So, sorry. Yes?
How do you bind your bead?
Yeah, so the question was how I bind the bead to the vesicle, right? So we use, the bead is covered with streptavidin, and we have some biotinylated lipid in the membrane. So it's like a glue. So you need a tiny attachment, because if the adhesion is important, you will never be able to form the tube. So you need a tiny, let's say a local attachment, and then you can form.
So you just put on one molecule of the...
No, a patch, a small patch. But we try to have it reduced to, let's say, less than a micron square, much less than that. Yes?
At the contact with the pipette,
yeah.
For me the curvature is even larger. Well, how do you do that?
This one, I mean, is very large compared to that, right? So basically, we often have something happening here, but basically we ignore it. We look at what is going on in the tube. And
this can blow up the membrane, just this curvature and the contact with the pipette.
No, because no, okay. So the question is why the vesicle is not exploding because of the pipette. So the pipette, we managed to treat the pipette to have no adhesion, let's say, between the vesicle and the pipette. So there is no adhesion, otherwise you could not aspirate.
Yeah, but which also means that the curvature can be...
Yes, no, it's a drawing. Okay, no, I agree. It's a drawing. So no, basically this is, for sure, this, but this is really not a drawing. You can see sometime when you move the pipette, there is some perturbation, but at the end of the day, when everything is stable, you have a nice tongue.
So, even on this picture here, what I saw, the curvature at the contact with the pipette is stronger than the curvature in vesicle.
You mean here? I mean the pipe, the
corner, the contact corner
here?
Here. So
Here, yeah.
Same corner on this picture.
Here?
Yeah, and the curvature is stronger here than the one you made.
Then here.
You see what I mean?
You mean that angle here?
Yeah, this angle, for me, the curvature is larger.
No,
I'm just even on
no, here, I mean, basically the curvature here, I mean, we are talking, this is very of the order of 100. This one, here. But this one, I mean, maybe it's strong or not. I'm not looking at the recruitment of protein there. So, but it doesn't explode the membrane. It's not that strong, that curvature. I mean, because the pipette, the glass is quite thick compared to that. I mean, no, the worry could be that you could have adhesion between the glass of the pipette and the vesicle, and this could create some explosion. But practically, I think that it's kind of smooth at that scale. Okay, so we can discuss this later. Anyway, so if we do that, so we have done experiments with this protein which were involved in the endocytosis at the late stage. One of them is called, you don't care, is this kind of very curved structure, N-bar protein. And what we have done here, you can see, is a condition where we have very little concentration of protein on the vesicle of the order of 1%. It's even not detectable when you have the overlay with a lipid. And you can see immediately that the tube is very green, meaning that you have a strong enrichment. And if you measure the density of protein in the tube divided by the density of protein in GUV, you can see that at very high curvature, and in this case, I'm talking about a tube radius of the order of 9 nanometer, we have an enrichment of factor 15. Okay, and you can, using the kind of coupling, linear coupling between curvature and density, you can predict that the enrichment that you can calculate should be directly given by this simple relationship, should be proportional to the curvature, 1 over RT, which is a curvature. And with the parameter here, which you can see is the parameter I mentioned before, the intrinsic curvature of the protein. And doing this kind of experiment at different density of protein on the GUV, and changing the curvature, etc., we find a spontaneous curvature of the protein equal to 1 over 7 nanometer. So we did that for this protein, which is also involved at the late stage of the endocytosis, and this one, which is clearly from the structure less curved. And we can see that at least in all cases, the theory was verified. You could see a nice correlation between sorting and curvature. And from there, we deduce a spontaneous curvature, which is very high for these two proteins and much less for this one. And at least for us, it was very encouraging because not only the theory was correct, basically we expected to get this linear variation of the sorting with curvature, but if you look now at, as I mentioned before, the neck of this vesicle when the protein are binding is of the order of 10 nanometer, which is basically the spontaneous curvature of the protein. So it explains why these proteins are recruited normally in the late stage of the process. Okay, so at least so far, yes, oh, yes.
I was expecting to have less phospholipid packing in the tube than in the tube, right?
Yeah, so your concern is a lipid packing defect in the tube. Yeah. Probably they are important, but they are part of the, they are somehow, they are somehow, in this CP bar, we have a kind of a very, it's a coupling coefficient. So you might take this into account somewhere.
So if you simply increase the tension of the GUV to make more defects on a flat GUV,
yeah.
Would the protein still bind efficiently as on tubes?
Yes, yeah. No. That means there is a clear difference between the curved structure and the flat membrane. And in this case, the tension is the same everywhere, right? So the tension is the same on the GUV in the tube. And what is different is the curvature. So maybe there is some additional defects which appear, which are for sure, but I think this is not the only reason why they bind. I think it's because of really the matching between the curvature of the protein and the curvature of the tube. Yeah.
And look at the recruitment coefficient, whether it's higher or
we will look at that. So if look only on the GUV as a function of tension, the question is the lipid. We didn't see any much difference on the density as a function of tension. So this is much less, very much less than anything that we can measure on the tubes. Okay, so after that, we were working on this bar domain protein, and there are two papers which were published by two different groups, and they stress that this protein, endophilin, that I just showed you was able to sense curvature at low density, was also involved in a clathrin-independent endocytosis. So in a process which doesn't include clathrin, which somehow involve the formation of tubules. And they showed that in this case, endophilin is probably involved at higher density, but more surprisingly, is able to induce membrane fission. So if you remove endophilin, you have much less fission. You still have fission by this protein, but much less fission. And then we were very surprised. We were very surprised because there were all data. You can see from that group, from Pietro De Camilli, showing that, again, if you incubate endophilin with a liposome, you see nice tube. You don't see fission, right? No fission. And more recent papers, same thing. So how is it? So we were doing experiments at high density, so to somehow mimic what was going on in this process. So what we were doing is now, we started from a vesicle. You can see here we have a large tube because the fluorescence is high, so it means that it's a large tube. And we are going to inject the protein, and you are going to see what is going on. So we measure the force. You see the protein is binding a lot on the tube, and you can see that the fluorescence on the tube is decreasing. So if you convert this fluorescence into a tube radius, you can see also that the protein, when it's binding, is also constricting the tube. And to give you an idea about the spontaneous organization of the proteins on the membrane, I mean, there was this work which was done by this joint PhD student between Greg Voss and me, Mijo Simonovic, who was doing kind of a simulation of the spontaneous assembly of the proteins. So the bar domain protein here, this is a coarse grain simulation. You can see the bar domain protein. So they spontaneously organize into an helix. And this helix is what is somehow constraining the tube. And now if you measure, so probably I was a little bit quick, but I told you that normally the radius should change with the tension. So if now you measure the tube radius from the fluorescence of the tube as a function of tension, if you have no protein, it should scale like here, in this curve. You have a square root of 1 over tension, right? So you should have a decrease. And you see that if you have endophyllin, you see that whatever you do, you aspirate your vesicle as much as you can, your tube radius doesn't change. And for us, it was the proof that the protein is somehow assembling, forming a scaffold, like a rigid scaffold around the tube. It also tells you that if you look at this structure here, you see that the force to hold the tube becomes 0. So it means that, in addition, the protein is stabilizing the tube, is not fissioning it. And the question is, how is it possible that in vivo they observe fission? So for that, there is a paper which was also published about the same time, and they also mentioned that when these tubes are formed for the endocytosis, they involve usually further in extension by some molecular machine called molecular motor that you can see here, which are somehow extending the tube when it's formed along this kind of rail, which is the macrotubules. So it means that the tube, it might be formed, but there is something pulling on it. In our experiments, what we do, we have pulled a tube, we have injected the protein, but the tube is static. So the question is, maybe we should pull on the tube and see what is going on. So this is the experiments that we were doing. So we have a vesicle, as you can see here, covered with protein, you see, on the tube as well. And now we are going to extend further the tube. And if you do that, so you can measure the force. You can see the force over time increases up to a point where the tube is broken. So it means that statically the tube is stable, but dynamically when you extend it, it's going to fission. And we get that, as you can see, for kind of very low, we can use very low extension rate of the order of 20 to 30 nanometer per second. It always works. And also, sometime we also observe that we can have, as you can see here, only a partial scaffolding of the tube. And you can see, in this case, the scaffold will be only on this part. And this would be different images, a sequence of images that we have taken. We start to extend the tube here. And you can see in this case that the scaffold is moving together with the vesicle. So it means that the scaffold remains connected to the GOV. So how can we understand this? First thing is when you have to keep in mind that if you want to form a tube or extend a tube from a GOV, you need a flow of lipid, right? And this flow of lipid will allow to balance the tension in the vesicle and the tension in the tube. And for instance, if you pull a tube from a bare membrane, as you can see here, this is a pure lipid membrane, no protein. You can see that you can pull the tube over over time, a different tension, as you can see here, or a different velocity, and you can see that the force is constant, right? So it means that the force is constant, means that the tension is constant, is equal, the tension in the tube is equal in tension in GOV, and there is no scission. So we were wondering if maybe there is a problem with the advection of lipids from the GOV to the tube. So we did two experiments. The first one, we were looking at the diffusion, how the lipids diffuse from the GOV to the tube. For that, what we did, we bleach completely. So we kill the fluorescence of the lipid in the tube. And if the tube will be normal, if there will be normal diffusion, you should get a recovery by simple diffusion through the neck in the tube. So if you do this when you have no protein, you can see that this would be a snapshot of the tube. You can see that after a few seconds, the tube recovers this fluorescence. And if you have the protein in the tube, on the tube, nothing. So it means that clearly the presence of the proteins somehow perturb the diffusion. And moreover, we can also look at advection. So for that, we use a trick. So we have a case where we have, you can see here, we have a portion of the tube which is covered by the scaffold of protein. We have a tube that we can measure the radius here by the fluorescence. And now what we can do is changing the tension in the vesicle with a pipette. So if would be no problem of advection, you will have what we can see here. There will be, so this will be something proportional to the tube radius. So here, if you have a control, no protein, you change the tension here and you increase the tension. You can see that the tension immediately is equilibrated in the tube. But if you have the protein scaffold, nothing happens. So this tells you that the presence of the scaffold somehow limits strongly the advection of lipid between the GUV and the rest of the tube, right? So based on this observation, yes?
Just a question, like, why is there less phospholipid signal under the scaffold? Is it because it's smaller, or?
I didn't explain it. So the question is, why is there less fluorescence here when there is a scaffold? So this is because the tube radius is there. I mean, you have a tube which has a diameter which is below the optical resolution. So it means that the direct fluorescence of the tube is proportional to the tube radius. So when you have a low fluorescence, it tells you that the tube radius is small. And we have even, so I didn't have time to show it, but we have calibrated the fluorescence to measure directly the tube radius independently of any model. So this tells you that the tube radius here is basically of the order of 10 nanometers, as I mentioned before, right? So, this somehow, together with Andrew Calajon, we came to the proposition that because of the presence of the scaffold, there is a friction between the endophyllin scaffold and the membrane, which appears when you are going to extend the membrane, right? And because when you extend the membrane as a velocity, to extend the tube at the velocity v, somehow you create a difference of tension because of this problem of friction between the tension in the tube and the tension in the vesicle. And this depends directly on the friction. So basically, you have created a friction. And since the membrane tension is directly proportional to the force, if we measure the force, we should be able to measure this friction. So we did this extensively. So we measured the variation of the force when we pull the tube at different velocity, as you can see here. And you can see that the faster you go, the faster the increase as you expect if you have a friction. And so Andrew was able to describe this extensively. And you can see that basically you have two regimes. Like in a viscoelastic material, you can see that at short time, the material behaves as an elastic. So you pull fast enough that there is no lipid which are coming into the tube. So you are pulling on the membrane itself. And after some time, there is a crossover. You get a flow, but the flow now, and you have a force which is plateau, and that force and the plateau of the force will depend directly, in this case, of the velocity with a power 1/3, and as well of the friction coefficient. So if you do that systematically on many experiments, you are able to measure a friction coefficient, which is okay, fine. And it's difficult to compare to something else, but what we can do is mutation. So the beauty of biology, you can do mutation. So you can perturb your protein. So for instance, we can create a mutant where we have less interaction with the membrane. So basically, we would expect a lower friction, this one. And we can make a mutation where the protein will interact stronger with the membrane. So then we expect a higher friction. It's precisely what we have measured, at least something which is consistent with what we expect. Less friction, more friction. Okay. So the question is, okay, we pull, we understand there is a friction, but how is the tube cut, right? How is it broken? So I'll remind you that if the tension of the membrane is increasing enough, it will reach a critical tension, which is the lysis of the membrane. So as I mentioned before, you can pull, but at some point there will be a tension which is high enough that will nucleate a hole, and this hole will blow up the system and break the tube. So if this is true, and it's very difficult to directly control that, but indirectly we can see we can predict how the force at the break will depend on the velocity. And for that, Andrew was using again a theory which was coming from Evan Evans, telling that if you create a hole at a different, let's say, rate of tension increase, you can you can incorporate into your theory this rate-dependent nucleation. And then from that, you can predict that the force break should scale as the velocity with a power 1/3. So we did that for all our construction. This is a wild type. This is a 2 mutant. We plot as a v and 1/3, and from the slope, in practice, you measure a friction, and we find a friction with this type of measurement as well, which is compatible with the first part of the experiment. So this somehow confirmed that at least the tube is broken because lysis in the tube. So to conclude, basically what I showed you, that to understand how these tubes are broken in the presence of endophilin, for that you need a protein which is creating a friction between the coat and the membrane. You need that the coat remain attached to the GUV, otherwise if you pull, basically nothing will happen. So you need somehow to have a strong binding. And then if you have that, the tension will increase up to the point that you reach the lysis and then you break. So this shows you something that we have also done, which is reconstituting a little bit more, in a more biological way, the tube pulling by molecular motors as it's occurring in vivo, by molecular motor moving along a macrotubule. And you can see also in this case that we get the tube also, which is scissioning in the presence of the protein. And we think also that this process that we call friction-driven scission is also an alternative of normally what is considered the canonical way of fissioning membrane, which is with this protein, which is dynamine. So the only thing you need somehow to have this process playing is you need a protein which is forming a scaffold, and you need a force. You need something which extends the membrane. So it could be a molecular motor, but it could be as well actin polymerization, which could also extend the tubule. So I don't know if I have time to go to maybe short. Huh? Maybe you have more questions. So maybe I will skip the rest, which is more about philopodia. But I think I will need more like 10 minutes for that. So maybe it will be too long. Huh? So I will just conclude, right? So I skip the philopodia, and it doesn't matter. I prefer to have questions and then okay. More 10 minutes. Okay. So I go to the conclusion. So I don't know, at least on the part I was able to present you, at least I showed you that to get this kind of approach, I think there are two essential points at least for me. At least you need first a collaboration with biologists, because you need to identify the right question and the right proteins to be sure you are able to build up a minimal system to address the right question, right? In addition, it's very good to collaborate with theoretical physicists, somehow to have at least to have some models that you can directly quantitatively test, as I showed you with this kind of tool. And I showed you also that, at least on the part I was showing today, that reconstituted biomimetic system could be a very useful and well-suited system for testing this hypothesis. So the problem that we are facing more and more, that when we try to address a biological question, we have to have built up more and more sophisticated biomimetic system. So this is tricky, more and more tricky. But I would say this is part of synthetic biology, which is a part of biophysics, which is somehow building more and more, and which is now growing at least a lot in Europe. Not enough, I guess, in France, but I hope it will develop more. And I would like to finish by thanking the people who were doing the experiments. First, the experiments on amphiphysin were started by Benoît Sor, who was a PhD student in my group, but now he's a senior researcher for the moment at MSc, and he will be back to Curie soon. So the endophilin experiment have been done by Mio, who is a postdoc at Rockefeller and just got a position at Columbia University in New York. So Colleen Prévost did the experiments of this. I didn't have time to show you, but she did experiments on philopodia, the protein involved in philopodia, together with Fong, who is postdoc in my group, who reconstitute philopodia. And all this work, at least I was presenting today, has been done in collaboration with Andrew Cala-Jones, who is assistant professor also at MSc in Paris 7. And I would like to thank all these people, so you can see many biologists here, but some physicists as well, who have been involved in this work and continue to be involved in this work for many years. And I thank you for your attention. And I hope you have many questions still.
I was just a little bit surprised that in one of your big slides at the beginning, you were talking about quantum dots. You were showing something, which was actually some visualization of experimental data, and you were putting a quantum dot. What do you mean?
So basically, it's something which is used now for many years. Maxime Daron passed last year. Is one of the person who was using a lot quantum dot to follow diffusion of lipids of proteins in membrane. So basically, we use a quantum dot, which is a tiny particle, which could be.
My question is why you call it quantum.
Why we quantum?
It seems to be quantum scale for me.
It's not, no, but the principle of emission of light is I think is more from quantum physics, but it's not a quantum. It's not the size which is quantum. It's called quantum dot because the origin.
It's emit light.
It emit light. So yeah.
It behave like an atom.
Yeah.
So yes, more like atom.
Yeah. So we use it as a tiny fluorescent particle to report on the movement of protein or lipids.
Why you don't call it fluorescent dot? Easier to understand.
Yes, it's the way it's used. It's called usually.
The wavelength is defined by the size.
Yeah.
So this is the confinement, the quantum confinement will define the wavelength.
Est-ce que vous pouvez lui donner le micro, s'il vous plaît?
I was saying that this is the size of the quantum dot that defines the wavelengths. Okay, by the fact that you quantize the wavelengths.
Sorry if I created some confusion, but it's traditionally called this way. No, but in fact, I could have tell a fluorescent particle, it would be fine for you, for sure.
Does the speed you need to pull the tube in order to provoke the fission depends on the initial tube radius?
Excuse me, I didn't understand the
yes.
beginning.
So I was with the scaffold.
Yes.
Does the speed at which you need to pull the tube to provoke fission depends on the initial tube radius?
The tube radius is always the same when you have a scaffold. The scaffold, as I showed you, whatever you do, the tube radius is constant, whatever tension, whatever.
But before you add the proteins, I guess you pull.
But then you can do, you pull the tube. You can start from a small, big tube. You always end up with a tube radius. This is a definition of the scaffold. The systems always, because the protein has this very strong spontaneous curvature, and probably the protein is rigid enough.
Yeah.
So whatever you do, you always end up by an organization which has a spontaneous curvature, which is directly dictated by the protein itself.
Yeah, but I guess my question is, when you pull at the extremity of the tube, you have some kind of reservoir of phospholipids.
Yeah.
And therefore, maybe the amount of phospholipids you have left kind of dictates the speed at which you need to pull to make sure.
So at the extremity of the tube, I think what the problem is more the reservoir. So, so the reservoir is the GUV, is not on the other side. But because when you pull, basically what you need is to get some. And if you have so you're right. Now if you have no advection, basically what you do, you start to use your tube as a reservoir, and for sure, I agree. So this probably tells you
which speed you need to. Yeah.
So but most of the experiment we have done, usually we have covered the tube completely with proteins. To get this fission. So most of the time we have covered the tube. So in this case, the tube radius is homogeneous. But I agree, if you have a part which is, let's say, wider, basically you can extend it more. So let's say your elastic regime will be, or a crossover between elastic and viscous will be at higher time, right? Let's say something like that, right?
And maybe more biologically relevant question. Could you imagine that, like for COP2 or COP1 vesicles, once they are assembled before fission, and because they are transported through microtubules also, can you imagine that maybe a stipular process could be involved too?
As we think, this kind of protein, so this kind of scaffold protein are found in different places, not only at the plasma membrane, they are found at the Golgi. So I'm not sure, there are now people working on COP1, I think it's COP1, where they have shown that you have you might have some protein involved, following the same type of process, right? But I'm not sure that the vesicles which are formed, usually to get that is very efficient in structure where you form tubules, right? Because to form a scaffold at the neck, there is not much space, right? And so I think there is often other processes which basically are efficient there. So I think this, in cases where maybe at the Golgi, maybe on the endosome where it's known that you get tubule, that this kind of, and you have also motors pulling on it, where you can have this kind of efficient process.
Yes, thank you for the talk. Following Rashid's questions, so for me, it means that the tube breaks, okay, if the advection is slower than the velocity at which you pull. Do you see such critical velocity?
Yes, I told you. Yes, I say that we have observed fission above 20 nanometers per second.
Okay.
So if you go more slowly, somehow you see an increase, exactly what you say. So, but the threshold is kind of slow. It's 20 nanometers per second.
Okay.
And if you compare to molecular motors in cells, so they move, these machines which are pulling the tube, they have been measured most of the time at micron per second. So I think we are fine.
Okay, and do you understand also why the coat is moving with the groove, and why don't you extend a tube in between the groove and the coat?
This I don't know, because we have no idea about how molecularly the protein are assembled between the vesicle and the tube. It's a good question, because of course if there would be no connection, you could do what you want and then will be no problem. So you need that connection, but we have no idea how it's made. And maybe to continue, maybe a little bit biology, so I apologize for the non-biologists of the audience, but it's maybe why dynamine is working with this protein together, because dynamine is polymerizing essentially on the tube usually, and there is not, when dynamine is binding to a membrane, it's polymerizing only on the neck or on a tube, never on flat membrane. So it's maybe why dynamine needs also in some cases the help of protein like endophilin or amphiphysin to get this kind of effect also playing a role. Because dynamine, in this case, is basically polymerizing essentially around the neck, but not on the flat part.
Okay, have you been able to measure the growth of this coat? Is it growing from the groove, or is it nucleating somewhere in the tube?
I mean, most of the time we have seen the nucleation of this coat at the neck, so next to the GUV. And we see a growth. So we didn't measure the growth. You didn't do that systematically. But we have observed that most of the time it nucleates from the neck of the tube, and in some cases at the other extremity. But at least it always starts at one extremity, never in the middle.
Okay. And I have a last question regarding the first part. So as far as understood, the bar proteins are recruited by the curvature of the membrane. Yep. Okay, so it means that you have fluctuations, spontaneous. You have spontaneous curvature. Then bar proteins are recruited, and so it means that the lifetime of the fluctuations is longer than the arrival time of the bar proteins, in order to make the process efficient. So my question is, what is the intracellular concentration of those proteins?
Normally the concentration is not that high, but so it depends. So what is important at the end of the day is how much protein you have on the membrane. So this can for sure depends on the bulk concentration. It might depend also on local, let's say the PEEP or whatever, which can increase the affinity of the protein for the membrane. So you might have cases where protein can be at a high concentration and working in a dense way, and in some cases not. But usually the concentration is not high. And there is only one type of bar domain which is at high concentration with a high affinity, is in your cell muscle, where you need these T-tubules, when you need a lot of tubular structure to have an extreme excess area to maximize the exchange of calcium to trigger your muscles. This is the only case, I mean, one of the rare cases where bar domain makes stable tubules.
So you show the result where you have the polymerization around the tube, right? Did I understand correctly? Is this a water soluble? So it's soluble in the liquid phase, the protein?
No, it's just around the tube. Huh?
But they polymerize in the tube, or they are all the time?
Outside, around the tube, outside.
So why they don't polymerize in the bulk?
It's a self-assembling. Basically, if there would be no membrane, it's not a polymer, it's a self-assembling. So meaning that if you dilute, for instance, the protein, they can be all separated, right? So if you have them, let's say, if it would be a real polymer, the best proof we have, let's say, it's a simulation, which has been done also by Mio. So what he did, he showed that if you have a membrane which has a low tension, you can have the spontaneous formation of this kind of structure, right? Like long linear structure. But if now you stretch a membrane, somehow you perturb the interaction between the proteins, which are the membrane mediated, and you completely break this kind of so it's not a polymer, it's a self-assembling.
So the membrane has an active role in the polymerization. That's what you're saying.
I would say in the assembly, because it's not. Okay. There is no direct interaction between the proteins.
Okay, so somehow the membrane helps the whole thing to polymerize.
Yeah.
Okay.
Normally they don't do anything
and okay, related to this, so you have, I mean, it's surprising that it's quite ordered, right? And the membrane is disordered. So
I would say.
Do you have any?
Why not? But at least it's you can say that the organization is coming from steric interaction in a sense. If you have high enough protein and they have all this, it will be like an enematic, right? They will somehow all be self-organizing simply by steric effect, right? Yes. So, and in addition of that, you might have some additional effect coming from the curvature itself, but they tend to somehow combine their affinity for the membrane and the organization on the structure. So I would say they are ordered like a nematic in a sense. It turned out that they do this polymer, but if you look at even higher concentration, so when people do this electron microscopy, so they have a very high concentration, they spontaneously tubulate, and if you look at the organization, you can see a very regular structure. So in this case, it's a very dense polymer, right, or polymer organization. But it's really something which is like a nematic. They will try to optimize their packing and the shape of the membrane at the same time.
Okay, so they do have an active role also in the membrane after the polymerization.
I would say that's on the moment, but we have no idea how the lipids are underneath.
So
really,
that's my next. So
it does something because the lipids do not diffuse anymore. So they do something. They do something. They might organize. They might be perturbing the organization. We have not looked at that.
Okay, thank you.
I just have a question about how you measure the radius with fluorescence intensity, because you're telling us that the density, the linear density, well, it can be stretched, the molecule, right? So if you stretch the molecule, I guess you also have a smaller fluorescent signal because you have a smaller density of fluorophore. So how do you make the difference between both effects, between the radius and the linear density of the
so what we look is we look at the density of lipids, right?
Yes.
And we hope that let's say the density of the membrane is not perturbed, right? So there is no direct effect. So the way we calibrate, so I can explain how we calibrate. So this is easy because we know relationship between the force we can measure, the tension. So we can measure, calculate the radius from this relationship between force and tension. We calculate the radius and we measure the fluorescence. So this is the calibration. And then, okay, so we have the relation that we both way. So and then what we do, we directly measure the fluorescence of the tube in the presence of the protein. We assume that the fluorescent lipid is not interacting with the protein as well. So this we know it's true, that we have tested and there is no problem. And in addition, we hope that let's say there is no other effect on the fluorescence of the lipids. I don't know what stretching you mean.
No, because you showed us a curve where you have some elasticity, right? Just a pure elasticity.
Ah, you mean on the membrane?
Yeah, of the tube.
Yeah, yes, yeah. But somehow it's when we do the calibration, this is already, in a sense, it's already there, right? Because we directly change the tension to change the radius, right?
Ah, okay.
So we change the tension, we change the radius, we measure the force, and we have the fluorescence. So it's already somehow incorporated there.
Okay, yeah, I understand.
Yeah, I hope I didn't miss this point, but in your fluorescent images, the length of the scaffold along the tube is, it doesn't cover all the tube. And this length, it depends on the concentration of the protein which you add, or it depends on some other factors.
So this is not always the case, but it's a case when you start from very large tube. So when you start often from a very large tube and you have a high concentration of protein, what happens is you start to nucleate at the neck, right? So you start to have a scaffold, and now you have a scaffold which is somehow perturbing the advection or the equilibration of the lipids between the vesicle and the tube. So what happens at some point, you have a big, I don't know, call it a big bunch of lipids, and they cannot just go through the scaffold to go back to the vesicle. So sometimes if you go from very large tube at the beginning and a high concentration of protein, we have this kind of let's say incomplete scaffold because of, in fact, the same effect which is creating the friction.
But what I mean, can you control this length?
No, no.
Okay.
Maybe if we work longer on it, we could, but we never work on it.
Just very short question. In your talk, you consider the endocytosis, but does it mean that the same proteins, like bar proteins and the dynamine and all this, they also work in exocytosis? Are these the same or not?
No, because it's a different topology. So when it's endocytosis, basically you form a neck, and the protein are around the neck. The problem when you form exocytosis, now the protein are inside the cell, right? So they are inside the neck. So this is the opposite topology. So it's another type of protein which are involved in this case of fission, and they are called escort protein.
No, I agree, but I mean, the topology is the same. You have either in the case of the endocytosis or exocytosis, you have this.
No, but the problem is when you go from inside, so your protein are always inside your cell. There is no way. In my vesicle, I can play with it. But in the cell, they are inside, right? So when you do exocytosis and endocytosis, you have the plasma membrane and you do that, right? So the protein will be outside the neck. Now, if you go in the other direction, your protein will be inside the cell and also inside the neck. So this protein, the same protein, they cannot work in the opposite topology. So it's not the same protein because they cannot bind on this kind of let's say in this case it's a negative curvature as opposed to endocytosis, which is positive curvature. So it's another family and even more complex. So we work on that. It's even more complex than the dynamine.
Okay, thank you.
Well, with this, let's conclude the session and thank Patricia again for this talk.